All right, we are here. Is my mic working? Yes, yes, it works. Welcome back to another day, another adventure, and we are going to talk about DNA repair and recombination. So I would like to insert dab, dab. Okay, so that one's working. So now, yeah, let's start with the single base changes, okay? So you have 4 plus 1-ish for single base DNA repair. And regarding some pathways, let's start with the base excision repair. So if you have deaminated cytosine first. It's recognized by the uracil DNA glycosylase that would cleave the bond between the uracil base and the phosphate backbone, and this would lead to a site that's missing its own base. Well, because the base was wrong. But after that, something will recognize the missing base, and that's the AP endonuclease. So again, first, we if there was a deaminated cytosine, that's recognized by the uracil DNA glycosylase. Uracil DNA glycosylase. That and then that would cleave it. And then you have the AP endonuclease, which would recognize the missing base and cut off the sugar phosphate backbone. So now you have a super clear gap. Phosphodiesterase would remove the remaining sugar phosphate group, and you have the clear single nucleotide gap now. So again, the deaminated cytosine is recognized by the UDG, uracil DNA glycosylase, and then you have the AP endonuclease, which would recognize the missing base and cuts the backbone. Phosphodiesterase removes the remaining sugar phosphate group there. And then DNA polymerase comes in to add the correct nucleotide. And then you have ligase, which just tapes it up. It seals the remaining nick in the DNA backbone. And that's really great. So that repair is the first pathway to fix the unnatural or missing bases. And the repair of depurinated and depyrimidinated bases begins at the same point. 1, 2, 3. How many things are involved in there? About 5. Okay. Uracil DNA glycosylase identifies that, oh, that's wrong. It will cleave it, remove it. So that's the UDG, uracil DNA glycosylase, and then you have the, who is it? Before the phosphodiesterase. AP endonuclease. Okay. AP endonuclease would identify that, oh, that's missing a base. It will cut sugar phosphate backbone there. Phosphodiesterase removes it. DNA ligase comes in and then adds the correct nucleotides, and then DNA ligase, you tape it up. You tape it. That's base exit number 1. DNA repair. And then you just how does the glycosylase how does it work? So it would just sit on the DNA and probe it until it finds the wrong one. So how does it do that? It would scan and flip them. So it scans the DNA strand for bases that are either abnormal or damaged, such as a deaminated cytosine, hypoxanthine, or uracil, because you're not supposed to have any of those in a DNA strand. And then it would flip it once it identifies the thing, and once the unnatural base is recognized or flipped, you just have the glycosylase leaving it. Okay. Number 2, you have the nucleotide excision repair. So earlier it was the base excision repair. repair. Now we have the nucleotide excision repair. Repair? Yeah, repair. It would target the distorted DNA caused by lesions such as pyrimidine dimers. Nucleotide excision repair. So first, it recognizes there is damage recognition where the DNA strand contains the distorted region because of the pyrimidine dimer. Because of that, it distorts the DNA structure and it is recognized by the repair machinery. Pyrimidine dimer is recognized by the repair machinery. Then you excise the damaged segment. So that's the excision nuclease. And then helicase comes in. It unwinds the DNA around the gap. Helicase unwinds the DNA around the gap to remove the section, the damaged single-stranded section. And then you just have DNA polymerase to come in, and then ligase. So it's roughly the difference steps 1, 2, 3, 4, 1, 2, and 3. Well, you recognize the pyrimidine dimer, the excision nuclease would come in and cut. Helicase unwinds and it will help remove it. Polymerase comes in and the DNA ligase would tape it. So that, and then in strand mismatch repair, pathway would begin by, the mismatch is recognized by MUTE-S. That is a protein that binds to the mismatched bases. The MUTE-L would then help coordinate the next step. So first MUTE-S comes in and binds to the mismatched bases in the newly synthesized strand. MUTE-L is also recruited, and then the whole segment is removed from the newly synthesized strand, and that is through the nucleotide excision repair. So this is just going into the very specific steps. And you now have the exonuclease activity, which excises it, and then you have the polymerase and also ligase. So again, the first part for strand directed repair is first you have the MUTE-S attaches to it. MUTE-L is recruited, which will help with the removal. After that, you have the exonuclease, which would cut, and then DNA helicase comes in and unwinds, which will help remove it. After that one, DNA polymerase would come in and add the new nucleotides, and then DNA ligase would just help seal it. And then now you have the third one, direct repair. Whoa! So first you have DNA repair, which is the base. What was the first one? That is, I need a pen paper. One moment. I'm back. I have paper now. Well, scratch paper, I mean. We always have paper. Funny. Okay, so the first one for DNA repair was DNA repair. Repair. One, it was the base, right? It was base excision. So for single base, you have base excision. And then next is the nucleotide excision repair. excision repair. And then now you have the direct. Like how that's written. I will make it into a map. You have the single base excision, nucleotide excision repair, or that's for the strand. And then now we have direct repair. So it's a direct chemical reversal of the DNA damage. And in that, you don't excise it or replace. For example, you have a pair, you want to repair methylated bases. Some enzymes, such as DNA methyltransferases, would just directly remove the added methyl group. And then that's where it would restore it. This process is very efficient because it's rapid. It avoids cutting and resynthesizing, and it's a rapid means of damage repair. And similarly, we have photo reactivation, which is another type of direct repair, which and that's where photolyase directly repairs the dimers caused by UV light exposure. And how does that excuse me, how does that work? Well, you use light energy to break the cyclobutane ring formed between the pyrimidine dimers and specifically targeting thymine. So direct repair, we have two versions of it. The DNA methyltransferases, which will just remove the extra methyl group and easier. And you have the photoreactivation, where photolyase directly repairs the pyrimidine dimers caused by what? UV light exposure, because we know that UV would induce dimerization. Okay. DNA methyltransferases and next is photoreactivation, which is photolyase. And then we have number 4, the brute force replacement by translesion DNA polymerases. What is that? Good question. Well, let's discuss it. So in translesion DNA synthesis, it would use translation polymerases that would bypass DNA damage, which would have otherwise stalled the normal replicative polymerases. And the way it happens is, first, our replicative polymerase would come across the lesion. It would say, "Ooh, I see it." The sliding clamp, we mentioned that earlier. It's part of the replication. It would modify itself. And when the sliding clamp modifies itself, that is a signal that the polymerase is encountering a block. So the regular polymerase is then released, and then you load the new DNA polymerase, which is a translation DNA polymerase. It would call in a backup, and the translation DNA polymerase would then replicate over damaged areas, but they're very error prone and might incorporate bases that are opposite the damaged sites. So the translation polymerase synthesizes DNA over the damage, allowing the replication to continue even though the DNA template is compromised. And then you just once it bypasses the lesion, the covalent modification on the sliding clamp is removed, and then the normal DNA polymerase can just come in. So how, again, what's the fourth one? It's brute force. Brute force replacement by translation DNA polymerase. Okay. So first, the normal DNA polymerase with its clamp would be like, oh, I see a damage. The sliding clamp would be, I'm gonna change form like Sailor Moon. And then it would let go of the normal DNA polymerase. It would call in back up, aka the translation DNA polymerase. And then after that, it would just brute force go over the DNA strand that's compromised. And after it finishes going through that section, the sliding clamp would go back to its normal form, and then it would let go of the DNA translation polymerase and then call in the normal one. The fifth, what's the fifth one? Fifth repair pathway, because it's a 4+1, right? Well, the fifth one, which is kind of unofficial, is that the DNA polymerase itself, the normal one, would proofread the newly synthesized strand. Proofread. Proofreads. Proofreads. Okay. So you polymerize it normally. So the DNA polymerase would have the P and E site, the polymerizing and editing site. The polymerizing site is where it makes the new strand, and then after that the new strand passes by the E part. It would check it for any repaired bases, and if a mismatch is detected, well, it would pause and use the proofreading function. You have the exonuclease activity now, which would remove that specific nucleotide, and then you would just resume the polymerization. So again, you base pair, and then you detect the error. That would halt elongation, because the mismatched DNA polymerase would halt it, because the mismatched base would prevent the polymerase from continuing. There is the exonuclease activity, the 3' to 5' exonuclease activity of it coming in. It chews back on the mismatched base, and then once the incorrect nucleotide are removed, you can just continue DNA synthesis. So we have a question. DNA glycosylases constitute an enzyme family found in all 3 domains of life. They can. It is a remove a purine or pyrimidine basis. Yeah, it won't add anything, and it won't remove a nucleotide. It would remove a specific base. So what is that? DNA glycosylase. In cells that are exposed to sunlight, the UV light can result in a covalent linkage between 2 adjacent pyrimidines in DNA. If not repaired in time, such dimers can lead to inheritable mutations. Oh, and we just have a specific sequence in the intense sunlight. How many dimers can form in the region? It's gonna be pyrimidine dimers. Pyrimidines would be, it's purag, P-U-R. Purine is A and G. Pyrimidine is C-T. In this one, we have a ton of T. There is a double T at position. I can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. I think 1. There's 1. That's an adjacent. T and C and T, T. if they're beside each other, that's possible. But around 2 in this one, I think, yeah. Also indicate each of the following lesions, typically repaired via base nucleotide, base excision or nucleotide excision. And your answer is okay. Deaminated cytosine or depurinated residues. That's the glycosylase. So it's base excision. Thymine dimers. Dimers might be nucleotide. Wait. Yeah, it is depurinated. I think that's just base excision. Thymine is UV. Thymine. Ouch. Yeah, nucleotide. And then guanine might be nucleotide also. Next, now you have double-stranded breaks. Usually the most important one. Double-stranded breaks are more crucial because it can lead to more grave results. So to fix double-stranded breaks, you have the NH non-homologous end joining and homologous recombination. So again, double-stranded breaks, you have non-homologous end joining and HR, homologous recombination. So for non-homologous end joining, what's that? What is that? So it's very efficient. It's used to repair the double-stranded breaks, but it can lead to the deletion of some DNA sequences at the break site because it involves processing of the DNA ends and the end joining. So you have random integration of the DNA fragments. It's very error prone, leading to small deletions and maybe loss of genetic info. So you have the Ku protein involved in it. Okay, Ku protein. Ku. It plays a crucial role in it because it starts with the co-heterodimers would recognize the DNA break, and then the co-heterodimers would bind to the ends, bind directly to the broken ends. It would call in its friends, the additional repair protein, and then there is limited repair synthesis over there because you have some small deletions, and then it would just help like with the ligation after it. So the main star is the Ku protein. And next, you have the second pathway, which is the homologous recombination pathway. And that is this one, you are repaired using a sister chromatid as the template. And this pathway begins when you process the 5 ends by the nucleases. Okay, so you have the nucleases swing off one section of over there at the top and at the bottom. It would prepare that step would next one would be alignment and exchange of genetic material. Wait, let's go. I'll explain it again. Yeah, that one. And that is more, it's more, what's this called? Exact compared to the other one, which is error prone. And so again, HR is more exact, but complex. There are more steps. Okay. Again, you have a double-stranded break happening, and then Well, that might be because of damage, radiation, or errors. The break is recognized. You have the MRE11 nuclease complex, which plays a critical role in recognizing the double-stranded break. Then, after that one, you just have MRE11 complex along with the SPO11, which induces the breaks during the meiosis. Okay. So again, you have the double-stranded break over there. The MRE11 and SPO11 would see it, recognizes the break, and then you process the DNA ends. Well, after you recognize it, the DNA ends would be processed by nucleases, so you have overhangs. So from the beginning, double-stranded break is there. You recognize the break with the MRE11 and SPO11. The nucleases would generate the overhangs. Then you have the REC-A protein, which will catalyze the strand exchange, going up and going down. And then DNA polymerase would just come in. You have the ligase. You also have the holiday junction going on. So yeah, it involves a lot more steps. Well, it was MRE11 and SPO11, which would recognize the break. Nucleases would chew off the area, so you have the overhangs. DNA REC-A would lead it to exchanging some of its strand exchange. But DNA polymerase comes in, and ligase also. Pol and ligase. So if we want to double check, so for single bases earlier, we have base exchanges, base exchange, which would just be you remove. Well, you have single and double strand breaks. For single stranded breaks, you have 4+1 ways to fix it. Double stranded, we have 2. Let's start with the first 4+1 of the single. Single again was base excision repair, where you have uracil DNA, it's the first one. It's the first. Base excision, uracil DNA glycosylase sees it. Endonuclease removes, endonuclease would cut, and phosphodiesterase removes, and DNA polymerase comes in, and you're done. Uracil DNA glycosylase. Uracil DNA glycosylase. You have AP endonuclease. And then phosphodiesterase. And then you have the DNA pol and ligase after. But for the second one, the nucleated excision, the mismatch repair, the whole strand, you have the mute S and mute L scanning it, and then helicase comes in. Not yet, wait. Remove, strand removal, exonuclease, yeah. Helicase comes in, and then DNA pol and ligase. Direct repair is just methyltransferase or phosphodiesterase. Photolase, it's photoreaction. Alright. And then you just have the proofreading. And then N-hedge would be non-homologous end joining versus HR, homologous recombination. That has the holiday junctions. So going back to homologous recombination, that would be, you have some specific steps, okay? You have the non-pairing interactions where the 3' overhang starts to search for the sequence. Once you find it, you pair the molecule with the other stuff. Helix nucleation happens because the paired interaction begins to grow and stabilize, and then you have the rapid zippering, the swift extension of the paired region. Yeah, starts off with triplet-wise probing. And then you have REC-A or RAD51 proteins, which would promote the crossing between the strands. How does it do that? Well, you have the REC-A protein binding to the single strand, and that would require ATP. After that one, it would help form the heteroduplex, and then ATP is hydrolyzed into ADP. You use it up, and then the invasion is complete. Voila. Okay. After strand invasion, so note, you have it can resolve the HR can resolve double-stranded breaks through pathways with either crossover or non-crossover. It would just depend on how the switch happens. Because if you have a specific crossing which would lead to double holiday junctions, then most likely when you cut it, some things will be exchanged. Okay? And also holiday junctions can migrate. You have key intermediates and homologous recombinations. It can migrate. So you have the ATP and ADP, right? Every time it migrates from a branch point, it would just go. So we have a question here. Which of the following repair pathways can accurately repair a double-stranded break? Yeah, A, base excision, B, nucleotide excision, that's no. C, direct chemical reversal, no. D, homologous recombination, yes. E, non-homologous end joining. It can, but it's not accurate. For which of... Wait, this is... I must sticky note this part. These are questions. Questions. Next. In the following drawing of a holiday junction that undergoes branch migration, cutting at which combination of the sites would generate a crossover? It depends. I gotta see the result, but maybe A to D. Okay, next, REC-A and RAD51 protein carries out strand exchange during homologous recombination. Indicate true and false. REC-A hydrolyzes ATP upon binding to the strand. Yes. REC-A forces the invading strand into a conformation that fully mimics the geometry of a long DNA. Whoa, it is true. That mimics the double. Okay. Sampling of a homologous duplex by the invading strand is likely to occur in triplet nucleotide box. True. The triplet nucleoside and the REC-A bound invading single-stranded DNA binds and destabilizes the homologous duplex to allow the sampling of its sequence. Yes, it facilitates it. Okay, so sort the following steps in the order that normally happens during the repair of double-stranded breaks by homologous recombination. And okay, so the steps: ligation, DNA synthesis using undamaged DNA as the template, DNA synthesis using original DNA as the template, release of the invading strand invasion, nucleotide digestion. So nuclease digestion would be first, and then strand invasion. The question is undamaged or damaged the first one? I think, invading strand is, okay, it will just be original first, and then undamaged, and then you release the invading strand, and then ligate. Let's just counter check. One moment. Yeah, so first, nuclease, strand exchange, you repair undamaged, releases, invading strand is released, DNA synthesis continues using damaged DNA. Okay. So, nuclease digestion, strand invasion, you use, what was it? Undamaged DNA first. You use undamaged DNA first, you release the invading strand, then you use the original, and then ligate. Julio. Then you ligate. Which of the following is not correct regarding direct homologous recombination? Okay, so A, loss of heterozygosity can occur if a broken chromosome is repaired using a sister chromatid. I don't. Wait. So that's true? Okay. Repair of double-stranded breaks by homologous recombination is favored. Okay, yeah, that's true. Homologous recombination can rescue broken or stalled. Yes. Excessive use of HR can lead to cancer. Yes, because they can like keep on switching. And E, low usage can lead to cancer. No. No, no. Because the D versus E would be fighting. Why does DNA repair matter? Oh, good question. If you don't repair, then you will have, you accumulate the mutations. I don't think that's good. So yeah, it ensures the integrity of the genetic material. So normally DNA polymerization, it's 1 in 10 raised to 5. If you proofread it, theirs is 1 in 100. Strand-directed mismatch repair is 1 in 10 raised to 3. 1 in 1,000. So if you combine all of those at the same time, it's 1 in 10 raised to 10. So it's much better. Smaller rate of mutation. And note, single base mutations really occur at a high rate. And DNA repair is crucial because single base mutations can occur really frequently. So in a 24-hour period, depurination happens around 18,000 times. That's a lot. It's a lot. Okay, depyrimidination around 600 and etc. So I have this one. DNA damage would, the next part would be DNA damage would delay cell cycle progression because DNA damage triggers a cascade of events and it would delay cell cycle so because it needs to repair itself first. Okay. How does it start? It's a good question. The first, you activate, let's say you have X-rays which did double-stranded breaks. ATM and ATR kinase activation happens. So ATM, ATR, and then the CHK1 and CHK2 kinases are then called. The CHK1 and CHK2 would phosphorylate P53. We know that. P53 becomes active, and it would bind to the P51 gene, because normally MDM2 would, wait, yeah, normally you have MDM2 and P53 together. If you ubiquitinate it, it would be degraded in proteasomes. But if it is phosphorylated by the CHOP1 and CHOP2, which was triggered by the ATM ATR because of the X-rays, MDM2 would be boom released, and then P53, you now have an active version. It would then bind to the regulatory region of P21 gene, and then the P21 is transcribed into mRNA, translated into the P21 protein. The P21 is a CDK inhibitory protein. So with that, the P21 would bind to an active G1 or SCDK and SCDK compilation or complex. It would inactivate it, and then that's why the cell cycle is halted. So from the beginning, you have an X-ray. It induces a double-stranded break. Double-stranded break would trigger the ATM-ATR, which would trigger kinases, which would then trigger the CHOP1 and CHOP2. CHOP1, CHOP2 phosphorylates the P53, which would release the MDM2 that's attached to it, and also you now have an active P53, which would bind to the regulatory region of the P21 gene. P21 gene is transcribed, mRNA is translated. You now have a P21 protein, which would bind to the complex of the SCDK and G/S1CDK, and then that's how cell cycle is paused. And defective DNA repair can cause diseases. So an example is the BRCA1, that's the breast and ovarian cancer. It is normally repaired by homologous recombination. But if it's not repaired, well, you see what happens. So we mentioned earlier, how does homologous recombination lead to mutations and crossovers or gene conversions, especially during meiosis? Well, it can actually be a good one, because there's exchange of genetic material through the crossover events, and they are important to generate genetic diversity that can lead to mutations if the exchange is not precise. Okay? And in meiosis, homologous recombination occurs during meiosis 1, specifically during the phase where the homologous chromosomes pair up and undergo crossing over. They align and cross over the thing, and boom, you get genetic diversity, because the resulting daughter cells are very distinct, are genetically distinct now from their parent cell. And in the process of meiosis, you generate the heteroduplexes, where mismatched alleles from homologous chromosomes are brought together. When mismatches happen, such as the red and blue genes of this specific gene X, you have a mismatch repair machinery which would excise it. And then the DNA synthesis would just fill in the gap. Okay. And then you have meiotic recombination, again, which would shuffle the maternal and paternal genes. And then that's why imperfect DNA repair maintains a fine balance between fidelity at the individual level and diversification at the population level. And we just have some specific. Oh, this one, I like this one. Because this page is DNA versus allele versus gene versus genome. So it's just a really good review. Okay, so what is DNA? DNA is deoxyribonucleic acid. It is the blueprint of life. What is an allele? It's a specific variant of a gene. And for each gene, you have like an individual would inherit 2 alleles from each parent. And you're like, what is a gene? A gene is a segment of DNA. So again, you have DNA, deoxyribonucleic acid, which is the blueprint of life. You have gene, which is a specific part of the DNA. Allele is just a version of it. For example, a human would inherit 2 alleles from each parent. And then the genome is just the whole set of genetic material, a whole set of DNA in an organism containing all of its genes. And a genome contains both coding genes and non-coding genes, and it has 23 pairs of chromosomes. What are histones? It would help pack and organize the DNA into within the nucleus. DNA wraps around it to form nucleosomes, and that's where acetylation and methylation comes in, because you can either compact the DNA or not. And yeah. And then chromosomes are long thread-like structures composed of DNA and proteins. Humans usually have 23, each containing a single molecule of DNA. Again, usually humans have 23 chromosomes. Each one is just a very, very packed version. And during cell division, was it? Yeah. It condenses during cell division so that it is segregated into daughter cells. And how is the flow of DNA? How does it happen for DNA condensation and decondensation? First, you have the long DNA. It's inside the nucleus. Let's say it's very relaxed. It allows transcription. And then you now have the histones. The DNA wraps around the histones to form nucleosomes. So you have the circle. DNA would just boom, like ikot-ikot over there. And now you have a nucleosome. That's the beads on a string structure. The histones help organize it into the chromatin, a more condensed form. So you have 2 versions of the chromatin. You have euchromatin and heterochromatin. Euchromatin is the loosely packed version. Heterochromatin is the tightly packed, and that is transcription cannot happen with a heterochromatin. But euchromatin, that is good. Gene expression can occur. But heterochromatin is important food also because, during mitosis and meiosis, it can just distribute it. So within each chromosome, you have many genes, and that would be the segment of DNA that codes for proteins or RNA. Yes. So that is, I think, for DNA. Hello. Ooh. Is it working? Why is it in a start? Is it still in a starting soon screen? I don't think so, right? No, it works. 안녕하세요. Okay, I think for DNA repair, that would be all. We shall return with another one soon? Later? Which one? Yeah, I shall return with a different one, okay? But for that one, that is all. Bye bye.